Okay, so in the last little bit of class, we're going to talk about cells and cell growth. So it'll just be a little introduction and teaser because we're not going to talk about it again for another week. Because we've got the midterm tonight and the midterm rewrite on Wednesday. So we'll talk about this again on the following Monday. And I hope everyone had a good reading week. But we've talked about enzymes and enzyme reactions. Cells and cell reactions happen very much the same way, except you have one thing that's, I mean, you can think of cells basically as fancy little bioreactors that have enzymatic reactions going on in them. But what's the one difference between cells and an enzyme reaction? Pretty basic question. I'm not asking for anything elaborate. You just have a big bag filled with enzymes versus a cell. What's the big difference between them? Sorry?
Time.
Maybe, maybe not.
The membrane had to go.
Yeah, but we talked about actually cases where you could have enzymes immobilized in spheres. So if you had an enzymes immobilized in a sphere versus a cell that's the same size, what's the difference between the two?
More enzyme?
Kinda. Not really. I mean, you've got a lot of other size reactions going on for sure in a cell.
Maybe if you were using the cell as an enzyme, it would have to expend some of the energy that it would usually use for its functions as an enzyme instead.
That's part of it. That's part of it. What's it expending those energies for?
Growing?
To divide.
Dividing?
So making more cells, right? So that's the big difference between a big bag of cells, or a big bag of enzymes and cells, is that cells make more cells, whereas big bags of enzymes probably just slowly deteriorate over time. So the big thing that we have to be aware of when we're talking about cells is what happens when cells divide. And that cell division actually becomes a major part of how we design bioreactors and understand bioreactors. So still you've got this cell that's a big bag of enzymes that's making some chemical reaction happen, and you have the advantage that it's very easy to filter out cells if you want to, or separate cells if you want to. But now we've got them dividing, and they do different things when they're at different points in the cell division cycle. So the first thing you want to think about is what are cells doing when they grow? So we've got here now substrate plus cells goes to extracellular products plus more cells. Okay. So here, this is actually called, and often actually the substrate now is not necessarily the direct substrate of the chemical reaction that you're interested in, but it's the limiting growth substrate in your medium. So you have to have all this stuff in your medium that's going to make whatever product it is, like when we talked about all the things that cells need to live, like nitrogen and phosphorus and all the many nutrients cells need. Those all have to be present in the medium. Some of them are really necessary to make your specific product that you need, but that specific product that you need is not going to be made if the cells can't be making more cells and living happily. So typically our limiting substrate now is not the substrate that is directly making the product that we're interested in, but is probably something like glucose or lactose, or what ends up being the limiting nutrient and nutrient product to make the cells happy and grow. So although you need those other substrates there in order to make your product, typically when you think about what we need now by substrate, it's the component in your medium that is limiting your cell growth. Things get a little bit more complicated because sometimes cells will switch what that limiting nutrient is as they But typically when we start talking about it, we'll have just one limiting nutrient. We'll think about it as having just one limiting nutrient that determines how fast your cells grow. Does that make sense? More or less. Okay, so that's called autocatalytic because, I'll just write that again, the sum of substrates plus, we're going to call X our cells, the concentration basically of our cells, goes to sum of products plus NX. And in fact, many times, the products are not, like say for E. coli, it's actually quite rare to have your products be conveniently extracellular. Oftentimes the products end up still being inside the cells. You have to harvest the cells, break them open, and that's how you retrieve your product. Okay. So if we're now looking at net specific growth rate, okay, we're going to basically define it as dx by dt. So that's your growth rate, but we're saying it's specific, so we're dividing it by the concentration of the cells that we've got there. Okay, so it's the amount of growth rate per cell concentration. So you can imagine if you have 10 cells there, the change in cells with time is going to be less than if you have 100 cells or 1,000 cells, right? Like you're doubling, so the doubling is your net specific growth rate, but your actual growth rate is going to change depending on how many cells are there. Does that make sense? So your net specific growth rate will tend to stay constant. Your growth rate will depend on how many cells are there. Okay. So again, more things. I said it was our net specific growth rate. And the net specific growth rate will depend on two things: our growth rate and our death rate, right? Because some cells are dying while other cells are growing, right? And in different parts of the cell cycle, the cell growth curve, you're going to have a different proportion of the number of cells that are dying and cells that are growing. Make sense? Sometimes they'll actually call it or define it by the cell number. Okay, so this is usually cell concentration based on mass. Sometimes you actually count the cells. And so if you're, just so that you know, the R is there if you've counted the cells, the net or the G is there if you've based it on cell mass. Okay, so that leads to the important question of, so this is all in hours inverse, is the units of net specific growth rate, even if you count it or not. Does that make sense? Okay. So how do you figure out how many cells you have, or how much cell concentration you have? What are the ways that you could figure out how many cells you have? You have you definitely need a well mixed sample. What do you do with your well mixed sample? Caitlin?
Flow cytometry?
That's actually a remarkably bad way to count cells. It's a remarkably good way to find out lots of specific things about each cell. But you have to do a lot of fancy things to figure out what your cell concentration is.
Oh, you can use chemocytometry.
Yes. Yes, so how many people have heard of a chemocytometer? Hands up if you have. Okay, so a few of you. So the words kind of give away what a chemocytometer is. Chemo relates to blood. Cyto means cell, and meter usually means to count. So chemocytometers emerged as a way to count blood cells. So if you're looking for concentrations of blood cells, then that could give you that information. Basically, all it is a really fancy slide. I'm drawing it really, really big. Okay, so a little microscope slide, that's a little microscope slide, I'm just telling you there. These are the parts you handle it by, so you touch the ends. These are actually little, if you look at it from the side, I'm gonna draw it a little exaggerated. They're little wells that fit a lot of extra volume. And what you do is you have actually quite a thick cover slip over the top of this. So you put your cells in there, and then by capillary action, they fill in the whole space between the wells. And because it's quite a heavy cover slip, and that's very precisely made, this space, there's a defined height to it. So actually what you're getting in your hemocytometer is a defined volume of cells. So the cells float around like that. And you don't want it too big because then you won't be able, they'll be beyond the level of focus. You don't want to have to focus up and down to get your cells. You don't want it to be too small because then the cells won't actually fit in that space. So they've been very precisely made so that it's a very specific space in there, so that you get always the same repeatable volume that cells can fit in, and that you can focus on through the microscope. And then within that space, there are these little, I'm gonna just draw it as 4 squares, these little grids. When you do 4LL, you'll see exactly how the little grids are set up. But essentially what you do is you count how many cells are within, the grids are laid out so you don't lose track of where you are, and then you figure out how many are in a whole grid area, and then you know how much volume is over top of that grid area, and you can say the number of cells divided by, I think it's 10 to the minus 4 milliliters is the concentration of your cells. So I'm pretty sure that basically in that, you've got 10 to the minus 4 milliliters is the total volume you have. You can double check it when you take 4LL, make sure your numbers are right. But that's a very straightforward way of doing it. It's got some advantages and some disadvantages. One advantage of it is that you can put if you're looking at mammalian cells, you can use a dye called trypan blue. And trypan blue gets excluded by whole membranes, and if there are little breaks in the membrane, even though the cell looks alive, if it isn't, then the membranes aren't complete, and the trypan blue can get in and stain the nucleus dark blue. So your dark blue nuclei are dead cells, and the ones that have only on the trypan blue around the edge of the cells are alive. So if you use a stain in conjunction with using a hemocytometer, then you've got a very simple live dead count. It's relatively inexpensive. It's something students can do in the lab easily. What are some downsides of doing that?
Is it accurate?
It's a sample, so you probably should be counting a number of different squares, not just one. Typically, the total number of cells should never be less than 100, and you should have at least 4 reps, so you can get some idea of the statistical reproducibility of it. There's some user error that's possible in it, particularly if you're not sure how to use trypan blue and you're using a stain like that, you're not sure whether that's a little piece of Kleenex or a cell, which happens, I'm not kidding. What other downsides are there?
It's not in line.
It's not in line at all, absolutely. And it's pretty tedious. You sit there with a counter and someone interrupts you in the middle. So you need the counter. I don't know if there's a Big Bang Theory where Sheldon was like counting neurons or something. I don't know if you ever saw it. And Amy interrupts him in the middle at like 237. He forgets, which is not Sheldon, but anyway. But that's, so you have a little counter, you try to remember which square you're on. There are potential user errors to it, and it's not in line at all. Most of the counting methods, I will also say, are in line typically. But it's not fast and convenient, mostly. If things are growing really quickly, it's gonna take you 15 minutes to do your count, and if things are growing really quickly in that 15 minutes, it might have doubled even, depending on what
What
cell
was the
type you're
absorbance stuff that we did with in the lab?
Sorry?
What was the absorbance using calibration curve absorbance is that?
Yep, so that's the really simple way. Oh, and I'll say bacteria, of course, are 10 times smaller than mammalian cells. So there are some more divided hatch marks that you can do bacterial cell counts, but it's even more difficult than the mammalian cells. So they're used, but they're a pain. Much more easy is what Carl just said, which was what you did with the yeast, right? So cells give a certain amount of turbidity to a solution. They interfere with light going through the solution. So you can, there's a reasonably good correlation between the number of cells and the light absorbance of a sample. So again, you want it to be well mixed. You want to make sure that that's always the case. Sampling is always important. But you can take it, just bring it, as long as it stays well mixed, bring it to a spec, and you can get a pretty good idea of what your cell concentration is. Any other, what are some problems with that?
It gets to the point where it's not like linear anymore.
Yeah, so if it gets really concentrated, then just because of how light absorbance works, it's no longer linear. Yeah.
You couldn't tell the live or dead. You wouldn't be able to tell if they were live or dead.
Absolutely. You could have a sample of all dead cell debris and a sample of cells, and they would look exactly the same. But of course, one would be happily growing and the other one would be completely useless.
If you leave it out for too long, can you contaminate?
Well, I mean, that's true in all cases.
Issue in this one.
Generally, most of the counts happen pretty quickly.
But if you
compared-
count, I'm sure, so kind of like right away, whereas like if you suspect, sometimes like if you leave it, I guess if you suspect right away.
Yeah, so typically you want to do your counting as fast as you can after doing your sampling, in any case. So even with the hemocytometer, you wouldn't put it in the hemocytometer and like, let it sit there, or you wouldn't even take your sample and let it sit there for a long time. Typically, if you do have to take a sample and let it sit for a bit, you put it on ice, so that you don't have any further cell growth or metabolism happening, and it'll keep the cells happy enough. It's not ideal, but if you know that you're going to need to have a break between your sampling and your counting or your analysis, you'll keep it on ice. But yeah, so the biggest problem with that, well, there are two problems. The live dead one is one. What about if you have really big fat cells versus really small tiny cells? Would you have the same, and if you had the same, if you ended up with the same absorbance reading, do you think you'd have the same cell count necessarily? Not necessarily. So between kinds of cells, you may end up with different numbers that you're looking for. But at different points in the cell growth phase, you may have changes in cell size. So as they're in their exponential growth phase, which we're going to talk about in a minute, you may have different cell sizes than they're in their lag phase or in their death phase, right? So you can't necessarily tell the difference between, you may end up with different absorbance readings without necessarily having different cell counts. So they're not perfectly correlated, that's a certain level for sure. And they don't tell you anything about live and dead. So there are similar ones to optical density. Anyone think of any other ones to use? That are just sort of also just gross cell counts or gross ideas of cell volume.
Ultrasound thing. Same idea as light, I guess, but those sensors too lively talk to them.
So there's
Or sound.
They tend not to be used with cells. If you use ultrasound, it can actually break up cells. They actually can be quite vulnerable to sound. So an easy one, if you deal with large quantities of cells, is you just basically spin them down in your centrifuge and you can see what the volume is of the cells. Okay, so I'll write this down. So we had flow cytometer. Right. We had optical density. So what I just described is packed cell volume. So you basically spin it down and you end up with a pellet, and you can see how big your pellet is. And that can give you an idea of what your cell volume is, or how much cells you have. Okay. And similarly, you can take that same pellet and you can dry it, and you can do dry cell weight. Okay. So both of those, packed cell volume and dry cell weight, give you sort of similar information to what optical density would give you. They give you a gross measure of how much cells you have total. When you do your fermenter labs next term, you're just going to, just as you did with the yeast labs, you're going to use optical density. It's really straightforward and convenient. And there have been so many people who have been working with, say, E. coli optical density, that they can tell you that the optical density between 0.4 and 0.8 gives you the exponential growth phase of E. coli. So that's where they're growing happiest and best, is when between those cell concentrations. So just by dint of long experience, that's a very easy way to do a very, very quick measurement that will give you a very easy idea of how much your cells are growing. Make sense? Okay. There are other ways, if you really care about what the viable cell count is, what are other ways you can do it? It's really, really important to you to get an accurate idea of viable cell count. Yeah.
They're automated cell counters.
So they're automated. Do you know what it means, Nicole?
I don't remember the name specifically, but I know like one of them where it's kind of like a Mon Oct, but it will run a sample in a minute, and it'll take, instead of 4, it'll take like 50 samples.
So, Coulter counter. You'll also come across in 4LL3. It's an automated cell counter again. It's based on electrical impedance as this, so it takes the cells just the same way as a flow cytometer does. It takes the cells through a narrow capillary that only permits one cell at a time to go through, has a defined flow rate, unlike a flow cytometer, and checks the electrical impedance through as anything passes a certain point. And clear, like medium, will have one electrical impedance, and a cell will have another. So it correlates an electrical impedance change of this much to a cell number. And so it'll very rapidly take those cells up, count how many interferences there are in the electrical impedance, and then it knows, has the flow rate go on for a certain period of time, figures out what volume that's been, and can figure out cell concentration from that. Again, typical Coulter counters won't tell you the difference between live and dead cells, because the impedance is about the same between a live and dead cell. There are fancy Coulter counters that can do some optical staining checks at the same time. You can spend more money on. It'll do it very rapidly for you, sort of much the same way as a flow cytometer will really tell you a lot about whether the cell is alive or dead, or lots of other things about the cells. But so they've taken some of that possibility and brought that into some fancy Coulter counters, not the ones that you get to use in 4LL3. So that'll just tell you how many cells or even particles that look like cells you have going through, but it's a very fast and rapid, relatively repeatable technique. Okay, any other thoughts? If you really want to know viable cells, like cells that are capable of cell division. And it's really important to get accurate numbers. What could you do? You must have done this in your microbiology class. They're giggles.
Is it the serial dilutions and plating?
Yes. So you do serial dilutions and you plate and you do colony counts because each individual cell should be able to make a colony if it's capable of cell division. You look shocked and disturbed.
It never works. It always comes back.
Well, that's not good. That means there were problems with your sterile technique.
Yeah, no problem.
But that is, so serial dilutions get the dilution down until you have very small numbers that are dilute enough that the cells should statistically be far enough away from one another that when you plate them out, they make individual colonies that don't merge into one another, and you can count each one that becomes a colony. You're not gonna do that in 4L3 with bacteria because it's not necessarily that important how many viable bacteria you are. You're gonna guess that they're mostly alive during the exponential phase. But you will do it more or less with mammalian cells because mammalian cells can actually be healthy but not capable of cell division. In some cases, it's really important to know how they can divide. So there you do serial dilutions. You put them in a plate, and in a week you count how many colonies are formed, and that tells you what the number of cells that were capable of dividing was. Obviously, that's an incredibly offline technique because you find out a week later what the viable cell number is. Okay, so I will see you again in 3 hours for the exam, and we'll continue talking